Hi everyone, welcome to week 3. So this week we are going to cover the building blocks of life. So just to review what we did last week, hopefully everybody's finally caught up from the big snowstorm that hit the weekend before. And you've not only finished topic 1 assignments as well as finished topic 2. If you're still behind, this is the week where you have to catch up. If you fall too far behind, you're never going to recover. So do take the time, if you've fallen behind, to catch up with all your missing assignments as well as complete this week's assignments on time. So this week, don't forget, once I've graded your topic 2 assignments sometime today, that you take a look, review the feedback that has been provided, go back and look through your fellow students' answers and make sure you check your answers with theirs. And then that'll help you prepare for our topic 2 quiz due by Wednesday midnight. So you still have something to do in topic 2, don't forget about it. I'll send out a reminder. So with topic 3, we're going to be taking a look at the building blocks of life. So that includes looking at the 4 macromolecules of life, so that's carbs, lipids, proteins, and nucleic acids, as well as looking at how we can bring in certain molecules, gases, chemicals, things like that into our cells and get rid of things that we don't need out of our cells and so forth. So we'll talk about things like diffusion and osmosis. Okay. So let's take a look at our due dates calendar. So this is always a good place to start. Get everything measured out in your calendar for the week. So we're going to be starting our Adigia reading. So that is topic 3 of our Adigia textbook. So for those of you that are doing the 2-week trial, now's the time. You got to purchase the textbook so that you get the rest of the materials. So review topic 3 in your Adigia textbook, and you'll be taking those quizzes throughout. You should also be working on your topic 3 assignments. So we have 3 of them this week. So T3S1 through T3S4. So you'll be working your way through that. All 4 assignments are due by Sunday midnight. Okay. You will also be starting to think about your first formal lab report. So we'll talk about that this week. You will want to begin. This week we're going to definitely collect data for it, but you might want to begin formulating parts of it. But just realize that the deadline to turn it in is not until February 7th. So you do have plenty of time to work on it. It's not due this week. So our topic 3, we always have this kind of summary video produced by Dr. Blackwell. So he goes through kind of a summary of topic 2 as well as an introduction into the information presented in topic 3, particularly the textbook. So always good to watch that. We have our reading assignment. So the reading assignment will be topic 3, the building blocks of life, in our Adigia textbook. So you should have access to your textbook and be reviewing it now. If you don't, you got to get that access code this week. So head over to the bookstore. And for those of you, notice there's no clicky thing to get the workbook pages. So the workbook pages we no longer provide for you. So you need to have purchased the workbook. So if you haven't purchased the workbook, you need to actually physically drive over to CCC bookstore and purchase it. And same with the lab kit. We need the lab kit this week for our lab portion. So make sure that you have all of your required materials today so that you can complete everything on time this week. Okay. So that's the reading. And then, I'm down here with reading. Then our student roles, I haven't changed these, so ignore all those names. None of these people are in our class. But just make sure that you're looking at your new student role every week, since they do change. So which number are you, 1, 2, 3, or 4? The group letter doesn't matter, because that's just something that you've been assigned in your participate or as a participant. So you don't have access to other groups. Okay. So So let's take a look at the material for this week. So our first assignment that we're going to be completing by Sunday midnight is the diffusion and osmosis assignment from our workbook. So with this assignment, with topic 3, by the way, we always start out in your workbook, you have your preparation objectives. So this is a good idea to take your textbook and be able to answer these questions before you start. So diffusion and osmosis. First off, you'll notice that this class assignment is a little different in that we have kind of a lab report set up. So this is getting you a little bit more familiar with how to construct a lab report, a formal lab report. So you can see things like the abstract, the introduction, question, hypothesis, and so forth. We're going to be taking a look at diffusion and osmosis in this assignment. You can read through the information, okay, but just realize that we're not doing this little demonstration, okay? So we don't actually have a dialysis bag and a beaker, and we're not performing this. So you can ignore all of that, okay? Starting on page 70, let's see, 70. Actually, starting on page, I guess 70. You can kind of read through this information, but just realize that you don't need to answer anything here because we don't have any demonstration. So starting on page 70, so complete pages 67 through 76. 67 you can't really do, okay? But on page 70 is where we can start taking a look at the difference between hypertonic or hypotonic and so forth, okay? So for questions 14 through 16, you're circling the answer or writing it in, okay? So greater, lesser, equal, so forth, for each of the types of solution. And then for 17, you're going to label which solution is hypertonic, which one is hypotonic, which one's isotonic. And then same with down here, you're going to draw a picture of what you think an animal cell will look like if it's placed in that solution. Do the same thing with the plant cell. Okay, so which one's hypertonic, hypotonic, and isotonic, and then draw a picture. Okay. And then here we're just taking a look at our blood cells. So with the blood cells, they should be, if you've ever seen a blood cell under a microscope, they look like little concave discs. And so based on that information, okay, so this is a normal cell right here. So which one of these would be, our red blood cells do you think were placed in a hypertonic, hypotonic, or isotonic solution? So what happens to those cells? So label that, okay? And then we have some kind of a check for understanding question. So based on that, so, what would happen if a child was given isotonic saline? What would happen if the child was given hypertonic or hypotonic? So what's happening to cells when we're placing those cells in these different solutions? So obviously, if you've figured this out already, you know that the isotonic solution is equal. So this is the one that our animal cells prefer to be in. So why can't we give a child hypertonic or hypotonic saline? So what would happen to their cells? Okay. And then we have our summary questions that kind of review what we learned in that section. Okay, so do make sure, and there are some kind of application type questions, so these are a little bit more tricky. So you might need to, again, look up in your textbook about the advantages, disadvantages of having a cell wall and how that relates to osmosis and things like that. Okay, so that is our first assignment. So you're just posting the answers to your assigned questions in the discussion below. With the second assignment, this is going to be a lab assignment, so you do need to complete the pre-lab first. Okay, so if you take a look at the pre-lab, basically you're going to be taking some vegetables and you're going to be placing them in different solutions. So hypertonic solution versus a hypotonic, and so forth. And then you're just going to let them sit for a while just to see what happens to them. Okay, so kind of note their structure, you know, feel them before you place them into the solutions, and then wait about an hour and then feel what they feel like afterwards. Okay, so that's kind of a pre-lab. Then you're going to be using the osmosis across the cell lab to complete pages 79 through 88. Okay, so this is basically looking at some microscopic images of cells under in different solutions. So we're working with plant cells here, an onion cell. So if you can see here, you can see the different structures. We definitely can identify this as being a plant cell because we have a cell wall. So it's like these little rectangles. Okay, so you can see even clearer what happens. Whereas animal cells, since we don't have a cell wall, our cells don't have a uniform shape like that. Okay, so you can see what they look like. And then finally we have our prokaryotic cell. In this case it's cyanobacteria. And so each one of these is an individual bacterial cell. So you can kind of see what they look like. And then finally within this document we have a couple videos of movement of liquid within a plant cell versus an animal cell. So you can see what happens when we place a plant cell in an isotonic versus hypertonic versus hypotonic solution, as well as a picture or a video of our red blood cells, what happens when we put them in isotonic, hypertonic, and hypotonic solutions. Okay, so you'll be using these videos and these pictures and images to help you complete our osmosis across the cell membrane lab. So as far as the information, you're going to be starting out just by defining these prefixes, and then use the pre-lab, the results from the pre-lab, to answer question 2. Since we're not doing any preparation of looking at things under the microscope, you can skip page 80. But you're going to be using the images from our supplement here to be able to draw pictures. Okay, so that goes in those boxes. And then again, don't forget to write a title, your magnification, and if a stain was used. Okay, so it should state the magnification and if there was a stain or not. Okay, so this one was stained with iodine, for example. This one magnified to 450. It's the same one up here. It's just been magnified, so it still was stained with iodine. Okay. So once you've drawn those pictures and so forth, you're going to move on to the animal part. So that's with the cheek cells. Okay. So there's the cheek cell. There's another cheek cell. Draw a picture. Write the information. Okay. And then finally our cyanobacteria. So that's this image right here. So you're going to draw that, write the information. Okay. And then with the rest of page 83, just remember that we're not doing a wet mat, of course. So you're going to be instead using the information from the videos to be able to draw those pictures. Okay, so what does the plant cell look like when it's exposed to isotonic solution? So kind of draw what those plant cells look like versus hypertonic versus hypotonic. And then you'll do the same with the animal cells. Okay. And then we have some summary questions. So this is just reviewing what happens to the plants and animal cells under each of the types of solutions. Okay. Once you've completed the lab, then you're going to be posting your answers. And then moving on to our third assignment. So with our third assignment, we're going to be looking at pages 89 through 96 of our textbook. So we're going to be focusing in on the building blocks of life. So basically looking at carbs, lipids, proteins, and nucleic acids. Before we get into that, we need to know what the bases are for that. So what are carbs made of? What are proteins made of? What are lipids made of? And what are nucleic acids made of? And they're all made of, they're built up from monomers. So we'll talk about, in this section it talks about how we can take a monomer, a subunit of a bigger molecule, and if we take two of them, they can undergo something called a dehydration synthesis reaction, where we remove water, we dehydrate it, H from one monomer and then OH from another, to form a polymer. Okay, so that's two monomers put together. So we can do that. So you'll kind of show that by drawing a circle around the H and OH that will be dehydrated, removed, and linking the two monomers together. Okay. We'll do the same for basically, or I should say the opposite. So in the opposite of dehydration, synthesis is hydrolysis. So if we have a polymer already and we're going to break it down into individual monomers, we're going to use a hydrolysis reaction. So hydrolysis, hydro means water, lysis means breaking. So we're going to use a water molecule to break apart the monomers. Okay, so that's basically the opposite of what we did here. As far as the molecules, the molecule labeled glucose, we don't have that available. Okay, so basically you're just going to be kind of visualizing on your own what would that look like. Okay, so think of glucose molecules, so you can look that up in your textbook. Glucose molecules, C6H12O6, so there's, you know, how many carbons, how many hydrogens, how many oxygens. Think about using your textbook, how that differs from fructose, okay, the structure of glucose versus fructose. So these are textbook kind of questions that you have to refer to, so we don't have a visual for you. So this is what we call a monosaccharide. Disaccharide, we're taking two monosaccharides undergoing a dehydration synthesis reaction to produce a disaccharide. And then finally we have our polysaccharides. So poly means many. Saccharide would be sugars. So many of the monosaccharides linked together. Okay, so we have three that we focus in on of polysaccharides. We have glycogen, that's found in our animal cells, particularly our animal muscle cells. It's a polysaccharide found there. And then the other two are found in plants. So we have starch. That's a storage molecule in our plants for energy. And then cellulose is a polysaccharide that makes up the plant cell walls. So based on that, you're going to answer the questions. Moving into another macromolecule, we have lipids. So think of lipids as kind of like the fats and stuff like that. So you're going to take a look at an example of this looks like a triglyceride. So you have glycerol and then a fatty acid chain and so forth. So here is our triglyceride. So you can see what happens when it undergoes hydrolysis and breaks down into glycerol and 3 fatty acids versus when we take the 3 fatty acids and the glycerol and we put them together to form a triglyceride. Here is an example of a phospholipid. So phospholipids make up our cell membranes. So you can see how we have our fatty acid chains. So that is why it's a lipid. And then we have a phosphate group attached. So see how they are formulated, where you have kind of the phosphate groups are facing outward and the fatty acids are facing inward. And think about why that is. Okay, and then moving on to our proteins. So proteins are made of amino acids. So the amino acid structure is the same depending, for any amino acid. We have an amino group and a carboxyl group attached to a central carbon atom. And then we have a side chain which varies depending on which amino acid we're talking about. So when we connect two amino acids together, and then we get a dipeptide. When we get more of them together, we get a polypeptide chain, which will fold into a protein. And then the last set of macromolecules are our nucleotides. So think of DNA, RNA, even ATP. So those are examples of nucleotides. So all nucleotides are the monomers that make up the polymer, the nucleic acid. Okay, so that is our example of the building blocks of life for our 4 macromolecules, so make sure you answer those questions and then post your assigned answers. And then the final project or assignment that we're going to be doing is an actual lab assignment. So this is basically one that you're actually going to manipulate hands-on. So hopefully it'll be a little bit more interesting for you. So you will need equipment out of your lab kit. So again, if you haven't purchased a lab kit yet, you gotta get over to the bookstore and purchase one in person, because if you get it shipped, it's not gonna get here on time. Okay, so make sure you get that done. So first things first, you will have a pre-lab for our concentration of and temperature on osmosis. So you will need to create some solutions. Basically, you're going to create a 10% sucrose, 20% sucrose, and 40% sucrose solution using some materials from your home, so table sugar, as well as distilled water. If you don't have distilled water on hand, you can use tap water. It's not going to change the results much. Okay, so get that ready, and then this lab that you're going to be taking data from will be used in order for us to write our formal lab report. So you'll notice there's a lot of reading here, and so I do talk more about how to formulate a lab report in a separate video, so I'm not going to spend time going through like what the title and all that stuff is, so you can watch that video instead. But I will pay attention to how to set up the lab. So in this particular lab, if you don't have beakers, so just use cups of the same size. It doesn't matter what type of cup. It could be a glass, it could be a paper cup, plastic cup, whatever. Again, you don't have graduated cylinders, so you can use a beaker to kind of measure. You will need your dialysis tubing, so that should be included in your lab kit. You'll have tap water. Again, if you don't have distilled water on hand, you can use tap water. You don't need thermometer, so don't worry about that. You don't need a hot plate ice or water baths for that. But you do need your sucrose solutions. Okay? So when you are ready, you're going to watch this video on how to prepare your dialysis tubing. Okay, so Dr. Blackwell will show you how to set up the lab. So watch his video, and then when you're actually performing the lab, you're going to take a picture of your dialysis tubes before you put them in the cups and after you remove them from the cups. Also, you'll need to include a selfie of you performing the experiment. So these pictures are going to be used in your lab report that's due February 7th, I said, something like that. So have those, somewhere. Okay, so don't forget to take pictures of yourself before and after the lab performing the experiment. So this is basically proof that you actually did the experiment, that you didn't just steal somebody else's data from another class or one of your fellow classmates and are claiming it as your own. So you actually have to, this is fun. It should be fun actually performing an experiment. So do make sure that you're doing the experiment. You will be posting your data for the concentration part of the lab, okay? But we're not doing the temperature part, all right? So let's go back. So once you've completed the lab, basically you're going to put your tubing into each of the cups. Each cup has a certain percentage of the sucrose solution in there. Okay. One more thing, does it say? I'm pretty sure it says you need to weigh it. Yes, okay. Just double checking. Before you put your dialysis tubing into the cups of solution, make sure you weigh the bags, okay? So that way you'll get the weight before you put it in, and then after the dialysis tubing has been sitting in the water for an hour, you're gonna take it out and weigh it again. So that way you can see, did it gain weight? Did it lose weight? Is the weight the same? All right, so do make sure that you're weighing them before or after. with a gram scale. So that was one of the required materials that didn't come in the lab kit, but you were supposed to have purchased or use from somewhere. Okay. So once you have your data, so you're going to fill out just this part, okay, because we're not doing the temperature one. So you are putting your sucrose solution. So remember, we made a 10%, a 20%, and a 40%. So that should be written there. But then what's this other one? So that's just the 0%, right? That's our control. You can write your initial weights of the dialysis tubing and then how much they weighed afterwards, and then figure out the difference in weight. Okay, so final weight minus initial weight. So if they gained weight, it would be a positive number. If they lost weight, it'll be a negative number. For this second part, since we're not performing the temperature lab, you will need to transfer the data from this link. Okay, so this is since we want to make sure that you don't have to do both experiments and collect data from that. We have the data for you. So you're going to take this information and you're going to transfer it. Okay, so you're going to transfer it over. So that actually goes, I'm sorry, not here, but here. So group 1, group 2. And you're writing down the difference in weight. Okay, so difference in weight for group 1, difference in weight for group 2 of the temperatures. And do make sure that you're writing down what the temperature was. Okay, so here we have 4 degrees, 25 degrees, 45 degrees, and 60 degrees Celsius. So here's the difference in weight for 2 groups. So that way you can kind of average the difference. All right. So once you have collected your data, you are going to be posting that data. So don't, even though this assignment is due on Sunday midnight, the latest you can post is Monday. Okay, you'll get a late penalty, but you can't post it anytime after that because students need your data in order to complete the lab report. Okay, so no, just post your concentration part of the lab. Basically, you are posting this part, okay, of your data. So this image, you take a picture of it, you can just write it down, whatever, but this needs to be posted no later than Monday midnight at the very, very latest. Okay, the rest of this is you'll be using in order to help you prepare for your lab report. Okay, so with the lab report, since it's not due until February 7th, you don't necessarily need to start working on it this week. I do recommend that you at the very least, take a look at my video, the instructor video, kind of go over it, and then maybe start thinking about writing the introduction and methods section of your lab report. So these two sections, since the methods section, you're describing what you did, you have that information this week. And then the introduction part, you can start looking at background information about that, which is included in our lab report, or our topic 3 lab here. So this information, you can kind of think about osmosis, what it is, and then why things will change the membranes, allowing certain things in and certain things out at different rates. Okay, so for this assignment, again, the only thing you're posting is your data, but make sure you post it, and then you'll be returning to this section when you write your lab report to get the data of other students to include with your own to figure out the results. Okay, so those are the 4 assignments that need to be completed this week. Okay, in addition to your topic 2 quiz that's due this Wednesday, and the Odigio reading that's due on Sunday midnight for topic 3. Okay, if you have any questions, feel free to post them in the Ask and Share you can email them to me, or if you need a little bit more tutoring help, things like that, you can set up a Zoom meeting with me. So just email me and we can set something up. Otherwise, have fun working on the actual experiment this week, collecting data, and I'll see you guys online.